Yeah, Dr. Sumit, have you joined? Hello, am I audible?
Yes, ma'am, I've joined.
Okay.
Yeah.
Okay. So I made you the co-host. Try to share your screen.
Yeah, I'll just do that. Yeah, can you see my screen, ma'am?
Yes. Just leave it at the first slide.
Yeah.
All right. I think we are good to begin. So good afternoon, everybody. And once again, welcome to the PG Masterclass. This month we have Dr. Sumit Rai from AIMS Mangalgiri talking on resistance in gram-positive cocci, mechanisms and detection. And if there are any questions, please put them in the chat and try to keep yourself muted while Dr. Sumit is talking. So Dr. Sumit Rai has been working very passionately in microbiology for many years, and he's now heading the Department of Microbiology at AIMS Mangalgiri. And over to you, Sumit, to take us through this class. And
Thank you so much.
just one disclaimer that this is being live streamed on YouTube. So in case you have any difficulty, anybody who's joining in through Zoom can please see it on our YouTube channel of IIMM PG Masterclass. And this is also being recorded for the YouTube channel. Yeah, over to you.
Yep. Thank you so much, ma'am. At the outset, I would like to thank Dr. Sonal and all the members of Indian Association of Medical Microbiologists for this wonderful opportunity for the teachers all across the country to get in touch with the postgraduates and deliver whatever the subject or the topics that have been allotted to us. Today, the resistance in gram-positive cocci, the mechanisms and the detection methods. So, yeah, okay. So I hope you see my slides changing. So this presentation is primarily going to be centered, as you guess, around Staphylococcus. And of course, we'll be talking about Enterococcus and pneumococcus as well. So as you know that the lot of resistance has emerged in case of Staph aureus and other coagulase negative Staphylococcus also. And MRSA is now present everywhere. We can't avoid it in any of the places or any of the new setups also like ours. We have got around nearly around 31% MRSA despite the hospital just being 3 to 4 years old. So it is everywhere. MRSA is present everywhere. And the typical part is that the resistance, especially to beta-lactams and even to the glycopeptides, is expressed in quite a heterogeneous manner. And epidemiologically speaking, the MRSA used to be, the classical definition used to be divided into community-acquired MRSA and hospital-acquired MRSA. And it has been very commonly seen that companion animals, like most of the strains and most of the isolates, have been acquired from animals and food animals and food products. And generally, basically, you can say the MRSA is the poster boy for One Health, just the way Salmonella is. So if you look at the timeline of the evolution of resistance, drug resistance in Staph aureus, you can see that the penicillin was discovered somewhere in the mid 1930s, 1940s, when it actually had a lot of effect. And subsequently, penicillin resistant strains evolved. For that, methicillin was introduced. There was multiple pandemics of penicillin resistant Staph aureus. Then, when methicillin was introduced later on, the first cases of methicillin resistant Staph aureus also developed. They had multiple isolates of MRSA in the US. And then eventually, after the starting of vancomycin, that was initially given as a very crude drug. And later on, now it's been much more refined. Vancomycin intermediate Staph aureus strains started evolving. And in 2002, the first cases of you can say, uh, the vancomycin resistant Staph aureus also started. And somewhere in 2000s also, 2000, mid 2010 also, we also had multiple strains of linezolid resistant MRSA also evolving. So we did have some studies of our own in linezolid resistant MRSA, and we also found the hidden resistance to linezolid in Staphylococcus aureus. So if I just talk about the brief outline, how we'll be going through the drug resistance in Staphylococcus is that we'll be talking about resistance to the beta-lactam drugs, including penicillinase production, MRSA, what is BORSA, what is MODSA, the resistance to the various glycopeptides, visa, H visa, VRSA, and what we call a slow visa, the inducible clindamycin resistance, the MLSB resistance, resistance to oxazolidinones, linezolid resistance, the hidden resistance to linezolid, and few things about this resistance in the small colony variants of Staphylococcus aureus. So we will not be dealing with the details about the epidemiology and the screening or the infections or the clinical features, treatment and management, prevention, infection control measures, for which this is a very good clinical microbiology review that I would suggest all the attendees can go through. This is a very interesting article they can go through. So we all know that numerous number of antimicrobial agents have been developed and are also in the pipeline for Staphylococcus aureus, of which many of them are the cell wall active agents. Some of them are acting on the protein synthesis, they are the inhibitors. Some are acting on the DNA gyrase. Some are acting on the transcriptomes, or sometimes they're acting on the metabolism. So they're all present, but resistance have been present in almost every class of the antimicrobial that is available against Staphylococcus aureus. When you talk about the gram-positive resistance mechanisms, we have to always follow the CLSI M100 document as much as we can. And there are 6 tables, at least 6 tables that we have to follow by heart. That is table 3G to table 3L in the M100 document that is now available. And also the many information, a lot of information you can find out in table 2C of Staphylococcus and table 2D in Enterococcus also. So these 6 tables, they are from 3G to 3L. The 3G starts from the detection of the beta-lactamase production by Staphylococcus species. Then we have oxazolidine salt agar test for detection of MRSA, vancomycin agar for Staph aureus and Enterococcus detection. Then we have got the table 3J for the detection of the inducible clindamycin resistance in Staphylococcus and Streptococcus and also beta-HS. And of course, the test for screening for high-level mucorosal resistance in Staphylococcus aureus, and the in the end, the HLAR or the high-level amine glycoside resistance for Enterococcus. So when you talk about the resistance to the beta-lactam drugs in Staphylococcus aureus, we have the penicillinase production, we got BORSA, MODSA, and the MRSA. So when it comes to the penicillinase enzymes or the penicillin hydrolyzing enzymes, we have to remember one thing, that overall, if you see in Staphylococcus, whether it's beta-lactam drugs or it is the glycopeptides, the killing mechanism is highly cell cycle dependent. So because beta-lactam drugs, they are the cidal agents, so they would be acting primarily on the dividing cells. So if we try and give any of the static drugs like erythromycin along with the cidal drugs, so the action of the cidal drug will get diminished. So we also should know that the different class of beta-lactam agents, they target individual penicillin binding proteins with different affinities. So there are a lot of variables which are present, and that's the reason why we see a lot of heteroresistance and variable expression of the resistance in Staphylococcus aureus. And in case of Staph aureus, the first and second generation cephalosporins have a greater affinity for the staphylococcal penicillin binding proteins as compared to the later generation cephalosporins. The gene which is primarily responsible for the expression of the beta-lactamase enzyme is the beta-lactamase Z gene that inactivates penicillin by hydrolyzing the critical beta-lactam ring. The method for the detection of the penicillinase enzyme is using two tests, out of which the penicillin disk zone edge test is the most sensitive test, and also we as available is the nitrocephin disk test. So the first table that we see, before we go to the table, if you can go through this diagram, this diagram shows that the most of the action happens in the periplasmic area where most of the enzymes are present, whether it's carbapenemases or whether they are the beta-lactamases. So you can see the beta-lactamase Z gene, it produces the beta-lactamase enzyme that is present in the periplasmic space. It has got an affinity for the penicillin and it inactivates the penicillin. The penicillin is acting through these porin channels. These are the outer membrane proteins or the OMPs. And similar kind of resistance you can see in Pseudomonas also when these porins they get mutated and they do not allow the entry of epenem, whereas they allow the entry of meropenem. So that's the OPRD mutation that we see in Pseudomonas. But in this case, the porins are intact. They allow the entry of penicillin inside, and the penicillin gets bound to beta-lactamase and it inactivates. So that's how they do it. So when it comes to table 3G, we can see the disk diffusion penicillin zone edge test, for which we are using any of the Staph aureus strains which MIC is in 0.12 or zone diameters of more than 29 millimeters. The medium that we use is oxazolidinone, and we use a 10 units of penicillin disc is used. It's a standard distribution procedure, nothing very complicated about it. The main thing is that the incubation temperature has to be 35 degrees, because if you increase the temperature to 37, then the heteroresistance does not get expressed very nicely. So for better expression, we have to keep the temperature at 35 degrees plus minus 2 degrees. When it comes to the interpretation of the result, what we see is that when we see the zone diameters, there is a sharp zone edge, that is a cliff formation if it is a beta-lactamase positive strain, or if it is a beta-lactamase negative strain, then you see a fuzzy zone edge that looks like a beach. So there's a cliff and the beach appearance is classically seen in case of the positive and the negative strain. So, and nitrocephin-based test can also be used, but then nitrocephin test has again to be confirmed by the penicillin zone edge test before the penicillin is reported as susceptible or resistant. So it is very penicillin susceptible strains, but ours being a new institute, we still do get maybe once or twice a month, we do get penicillin susceptible strains. So this is how it looks like. The plate on the left side, you can see the zone is absolutely very sharp. The zone is very sharp. It is an immediate end of the growth, and there's an immediate beginning of the no growth region. Whereas in this case on the right, you see there's a beach, there's a slope, there's a small, there's a slant kind of thing. There's a gradual fading of the zone of growth to the no growth. So you don't see any exact or a pinpoint edge. You don't see. This is a negative isolate, and this is one that is positive. So the penicillin zone, the penicillin disc zone edge test is much more sensitive as compared to the nitrocephin-based test. So if you have to do one single test to detect the penicillin's production, so instead of going for nitrocephin, you can go in for the penicillin disc diffusion zone edge test. And for these cases, if the patient is showing that the isolate is showing penicillinase activity, in this case, you have to go in for the penicillinase resistant or the penicillinase stable penicillins like cloxacillin, dicloxacillin. You can also go in for nafcillin or the first generation cephalosporins. When it comes to beta-lactamase enzyme detection, the penicillin distribution zone edge test is the gold standard test. Now, what choices do we have for the penicillinase producing beta-lactamase positive Staph aureus isolates. These are MSSA isolates which are beta-lactamase producing. So the options are, we can go in for oxacillin or nafcillin, which are the penicillinase stable penicillin. But the issue with oxacillin is that it has got hepatotoxicity. Nafcillin has got nephrotoxicity. Cefazolin is a first generation cephalosporin that can be given, and it is to be given every 8 hours, so that dosing is also not an issue. But the issue comes with the cefazolin inoculum effect. That's why the source control is the core and central to treatment of all the MSSA infections. And if you're giving any of the penicillinase stable or the penicillinase resistant beta lactam antibiotics, in that case the the source control has to be there. The inoculum has to be decreased. Third generation cephalosporins are not very superior for the treatment of MSSA isolates, and is one of the worst choices to use for MSSA. Why? Because I'll just show you one example of this. One of the reports. This is an AST report from our department that has been generated by HUNET, and then we customize this particular report as per the patient, where the patient is from OPD or from the IPD. So this patient is an IPD patient from surgery department from which MSSA was isolated, which was D test negative, and it was an MSSA. And we have written down also in the next slide, you can see what is the interpretation of MSSA in the advisory footnotes. So we say that we should avoid the use of oxacillin, nafcillin. We are doing it because of these reasons, because oxacillin has got hepatotoxicity and nafcillin has got nephrotoxicity, and therefore we are writing over here that MSSA isolates are likely to respond to beta-lactamase stable penicillins like cloxacillin, dicloxacillin, and flucloxacillin, and MSSA will also respond to BL-BLA combinations, but it is advisable to best avoid such combinations due to detrimental effect of oral BLIs on the gut flora like coamoxiclav. We're also saying in this case that not to use the glycopeptide drugs for MSSA, because the clinical response to glycopeptides is poor in MSSA as compared to MRSA. Now, can we give this? Can we give glycopeptide vancomycin for MSSA? So if you see, compare vancomycin to the penicillinase resistant penicillins, these penicillinase stable penicillins have decreased. They are able to decrease the mortality for MRSA bacteremia. There are known studies for that, and these penicillinase stable penicillins, they reduce the MSSA bacteremia also, and they also reduce the recurrence of bacteremia. And there's a more rapid kill of MSSA by the PRPs as compared to vancomycin, and they have an additional mechanism that improves the efficacy of the host immunity against MSSA compared to vancomycin. So that's why you should always avoid giving vancomycin for MSSA. So what about the inoculum effect in MSSA? So we have heard about the inoculum effect in case of ESBL infections, that if you got low inoculum ESBL infections, maybe we can go in for carbapenem sparing therapies, but for high inoculum ESBL infections, we have to go in for carbapenem therapy. The same thing applies to the inoculum effect in MSSA also. When the number of bacteria at the site of infection are when the number of infections in the site of infection are more than 10 per 7 CFE per ml, in that case, the hyperproduction of the beta-lactamase, it kind of destabilizes the penicillinase stable penicillins and even the cefazolins. And cefazolin, in such cases, there is increased production of the beta-lactamase production also. And so these kind of infections, they have got, you can say, the BORSA subpopulations also present. And these kind of infections, the high inoculum MSS infections, can be controlled by the combinations. And probably this is one of the success for drugs like Augmentin, when people think that even without the draining of the site or draining of the abscess, or even without doing the source control, it acts like a ramband and it works for those cases. So the ultimate aim should be that you have to reduce the inoculum and then start the antibiotic accordingly. It's a golden rule in all the infections. Talking about the BORSA or the borderline oxacillin resistant Staphylococcus aureus. Now there are gray zones between MSSA and MRSA. MSSA means it is penicillinase producing Staph aureus. Means it's an altered target site producing Staph aureus. So the resistance is extremely heterogeneous, and there are multiple gray zones between these two extremes, MSSA and MRSA. And what happens with BORSA is that there's normally hyperproduction of beta-lactamase enzymes. There are other mechanisms which might be present, but the most validated reason for the presence of the BORSA is the hyperproduction of the beta-lactamase enzymes. And initially, it was found that BORSA isolates may have oxacillin MICs of 2 or less than 2, and they're normally susceptible to penicillinase-resistant penicillins. And as we know that salt, because Staph aureus is a salt-loving bacteria, and that is the reason why it is present in the axilla, in the groin, in the nares where there is excessive sweating. So the beta-lactamase synthesis is enhanced by 5 to 10% NaCl. So whatever methods that we'll be going through, in practically every method, you'll be having 2 to 4% NaCl in all the media. So BORSA strains, they with the higher oxacillin MICs also exist, which may be resistant to penicillinase producing or the penicillin. And therefore, because they are beta-lactamase producers, they can be neutralized by clavulanic acid or sulbactam also compared to MRSA. So the prevalence is not very much known in Indian studies, but in the Western study, it is said to be around 5% of all the MSS isolates. The testing of BORSA is very, very challenging because there are no absolutely defined guidelines for the diagnosis of BORSA. But there was this very interesting article published in ASM Molecular Biology article that defined that what was the BORSA can be defined as. So BORSA, they say, is basically negative for the MEK, negative for penicillin binding 2A also. It's negative for that. It is resistant or intermediate to oxacillin by distribution when the zone size is less than or equal to 12 millimeters. So yes, oxacillin as a disc is being used occasionally, but in not all cases. And of course, if there is resistance to oxacillin by the E-test using 2% NaCl. And if there is consistently there is a MIC of 4 or more than 4 when tested in triplicate. So triplicate reading of MIC of 4 on a 2% NaCl agar for oxacillin and zone diameter than equal to 12 is suggestive of presence of a BORSA isolate. Then we come to the MODSA or the modified Staph aureus. In this case, the resistance is in Staph aureus. This is non-MEK-A and non-MEK-C mediated. So it looks like Staph aureus, it looks like MRSA only, but there's no MEK-A, there's no MEK-C present. Resistance-wise, it is above BORSA, but you can say it is below the MRSA. The MODSA isolates, they've got numerous mutations to the native genes, so which are encoding the penicillin binding proteins, the GDPP or the cation multidrug efflux transporter ACRB. So there are mutations which are the usual mutations. So BORSA is producing beta-lactamases. MRSA is a penicillin PBP2 positive, whereas in this case, PBP is being mutated, but in different sites. So this MEK-A and MEK-C is negative. Oxacillin MICs are high, and many times it's wrongly identified as MRSA. So if you start doing the penicillin, you can say the latex agglutination test or the MEK-A gene for the MRSA isolates, you might find some of them are negative for MEK-A and MEK-C. Those isolates are the MODSA isolates. So this was another very interesting study that defined the MODSA isolates as these are associated with unique truncating mutations in the gene encoding the cyclic diadenosine monophosphate phosphodiesterase enzyme or a non-synonymous substitution in the gene encoding PPP2. So basically the unconventional mutations in the PPP2, they give rise to a MODSA isolate. So if we talk about the from penicillin to MRSA, if we talk about from penicillin susceptible Staph aureus to MRSA, you can say they are like the 50 shades of gray. So if we begin with the penicillin susceptible Staph aureus, in this case, this is a wild type phenotype. That means pan susceptible to all the antibiotics. Then came the MSSA with an oxacillin MIC of less than 2, in which case the beta-lactamase Z gene was responsible for resistance for the beta-lactamase production. Then came the BORSA with an oxacillin MIC varying from 4 to 16 microgram per ml because of the hyper beta-lactamase production. Then came the MODSA with oxacillin MICs definitely than 4 microgram per ml because of the mutations other than MEK-A and MEK-C. And finally came the MRSA with oxacillin MICs of more than 4 with definitely the target site alteration by the presence of the MEK or the MEC gene. So that is the relation between the penicillin susceptible Staph aureus, the MSSA, BORSA, MODSA, and the MRSA. So that's how it has evolved over the years. So if we compare this in a table, the BORSA isolates and MODSA both are MEC-A and MEC-C negative, whereas MRSA, almost all of them are MEC-A positive. Some isolates might be MEC-C positive. In BORSA, there is hyperproduction of the beta-lactamase, and we can use the nitrocephin test because beta-lactamase is being hyperproduced, so nitrocephin will definitely be positive. In MODSA, like I said, they are truncating mutations in different sites, which are not normally found in the PVP2A. And in MRSA, of course, because the altered penicillin binding protein, the target site is altered. The BORSA isolates, they are inhibited by clavulanic acid, whereas the MODSA and the MRSA are not. So that's why this is the reason why the MODSA isolates might get confused with the MRSA isolates. When it comes to the testing by cefoxetine and oxacillin, cefoxetine is susceptible in BORSA, whereas oxacillin is resistant. In case of MODSA, both are resistant, and of course, MRSA. If it is cefoxetine resistant, oxa resistant, then it's MEC-A. If it is cefoxetine susceptible and oxa resistant, then it is going to be most likely MEC-C. So this is a very interesting thing that we must see, that what is MEC-C? MEC-C may, in this, you'll see cefoxetine susceptible and oxa resistant can be found. Latest agglutination of PCR for MEC-A and MEC-C will be negative both in BORSA and MODSA, whereas it's going to be positive in MRSA. And the BORSA, they lack the PVL toxin production. It's variable in MODSA, and in case of MRSA, especially the community-acquired MRSA, produces a lot of the PVL toxin. One interesting thing about BORSA is that identification might be an issue because it may lack the classical enzyme coagulase or the thermonuclease. It may be absent in BORSA, whereas the others, they express it properly. So in total, if we talk about the molecular surveillance of MRSA, MSSA, BORSA, MODSA, MSSA, and everything, so if we talk about Staph aureus, all these methods would be applied if you want to identify these, like proper identification of species by Malditov, the MEC-A and the MEC-C detection by the PCR, penicillin binding to A detection by the lateral flow, latex agglutination, drug susceptibility testing by cefoxetine distribution or oxacillin distribution, or of course we have their MRSA screeners are also present. Then beta-lactamase activity using looking at the penicillin zone edge this test, and also the nitrocephin test. And of course, in the end, we have to go in for the whole genome sequencing also. So these are all which are required for the analysis to distinguish between what are MSSA, BORSA, MODSA, and MRSA. So coming to the introduction, we know that the MRSA is basically the target site alteration is there. All the beta-lactam drugs, they are defined by 4-member beta-lactam ring that is essential for the biological activity. The cell wall of the gram-positive cells of the gram-positive bacteria is composed of the glycan chains of N-acetyl glucosamine and N-acetyl muromic acid they are cross-linked by the pentapeptides. And these precursors, they are synthesized in the cytoplasm, and the transpeptidase, they transport these precursors towards the division spectrum into the actually cell. And penicillin over here, it inhibits the cross-linking of the peptidoglycan by acting as a substrate analog. So instead of the transpeptidase, penicillin does so these transpeptidase, they are blocked by the binding of the penicillin. So these transpeptidase are called as the penicillin binding proteins. So the binding of the beta-lactams to the penicillin binding proteins leads to formation of long-lasting acyl complexes, and they block the transpeptidase activity, and therefore the peptidoglycan is not formed properly. So like I said previously, the killing mechanism is highly cell cycle dependent. How did MRSA actually emerge? So like I said, that the Staph aureus is one of the poster boys for One Health, and many of the strains of the coagulase negative staph, now it is known as Staphylococcus other than Staph aureus. So SOSA is the new terminology for cons. So it was said that the first strain of Staphylococcus aureus that gave the MEC-A gene to the to the Staph aureus isolate, and that became the first MRSA isolate. So through the mobile genetic elements, Staph aureus resistance to methicillin and many other antibiotics through plasmids or the chromosomal cassettes or the pathogenicity islands, or of course the bacteriophages. So all these methods are there. out of which for MRSA, the SCC-MEC or the staphylococcal chromosomal cassette, that's the most important. That is carrying the MEC-A gene, and this gene is present on the family of structurally complex genetic elements, which are being designated as the staphylococcal chromosomal cassette MEC. This is also a pathogenicity island because it's often carrying multiple genes which are conferring resistance to antibiotics also. So it has been found originally in Staphylococcus, which was present in the domestic animals, and the MEC-C, which is encoding the transpeptidase enzyme, with has got 63% identification to MEC-A, which is encoding the PVP2A. So both MEC-A and MEC-C have got the animal origin. So OH means the One Health. So basically, it is all related to the animals only. So we are getting all the Staph aureus, the MRSA, from the animals only. So far there are different, around 11 type of staphylococcal chromosomal cassettes have been found till date, out of which the 4 and 5, they are found in the community-acquired MRSA. So even though the definitions and the lines between community-acquired MRSA and the hospital-acquired MRSA have been blurred, but still, broadly speaking, community-acquired MRSA are usually causing skin and soft tissue infections in the community, and they are often fomite transmission is present and usually susceptible to many of the oral antibiotics. And the genome is smaller for this SCC-MEC because it is carrying the type 4 or type 5 SCC with the MEC-A gene. And most of the community-acquired MRSA strains, they carry the PVL toxin. And it is said that the community-acquired MRSA greater PVL toxin as compared to the hospital-acquired MRSA, primarily human-to-human spread inside the hospital. So these are different, just to enumerate the different penicillin-binding proteins which are present. Like for example, PBP2A confers methicillin resistance in Staph aureus. PBP2X, it confers penicillin resistance in Streptococcus pneumoniae. PP predominantly 4 causes the resistance to the beta-lactam drugs in Enterococcus fecalis. PP5 expression primarily is seen in Enterococcus faecium, which imparts resistance to the cephalosporins and even to carbapenem. So if we come back to the original diagram, again the same thing, but the difference is that over here we have got the altered penicillin binding protein, that is the PBP2A. So what does the MEC-A gene does? The expression of the MEC-A leads to the expression of a protein known as the PBP2A protein, and this protein has got a binding site mutation. So it does not bind to the beta-lactams at all. So there is no binding on the beta-lactams, so there is no effect on the transpeptidase activity. So transpeptidase activity remains intact, and therefore the cell wall is continues to get produced. So there's no effect site alteration, and therefore no matter what or what amount of the beta-lactam drug you keep giving, since the binding site is totally gone, so that is why we can afford to extrapolate the result of an MRSA from one to all the beta-lactam drugs. Like for example, in 2010, the issue regarding the ESBLs was removed. That means you could not extrapolate cefotaxime resistance to ceftaroline resistance because it is an enzyme-mediated process. But in this case, this is a target site mediated process. So that's why if the target site is gone, then even if you give coamoxy per generation cephalosporin, nothing will work. So if it is an MRSA, that definition can be extrapolated, that if it is MRSA, it is pan-resistant to all the beta-lactams, BL-BLI combinations. So this is an obsolete and old method, but still just for the sake of history, I'm showing you this thing, that when they use oxazolidinone disc, because I told you that the expression of whether it is beta-lactamase enzyme or whether it's a vancomycin resistance or whether it's the expression of the MEC-A gene, all of them are very, very variable. They're highly variable, and that is the reason for the resistance in case of Staph aureus. Now, what is happening over here? The use of oxazolidinone, it's a very stable drug, but because of heteroresistance, you see a fuzzy zone over here, which shows the heterogeneous expression, and in this case shows the homogeneous expression. So we don't want to see these kind of things because we want to express everything. We don't want heterogeneous expression. We want all these cells in the entire clone to express the gene, so that we don't see this kind of zones and create confusion. We have got an absolute 0 or 1 kind of interpretation. So if you see 2C of Staph aureus, the methods or targets for detection of methicillin resistant Staph aureus. So they're doing the cefoxytin disc for which the time of incubation is 16 to 18 hours. Now, the oxacillin is not used in the disc diffusion. Because of this reason only, this is not used. That is why I told you that the previous method was obsolete. And then again for the MIC, we have got cefoxytin and oxacillin. So oxacillin MIC we can do for all the different staph loci that you have, but the incubation time is 24 hours. Whereas in case of cefoxytin, the time is much lesser. So cefoxytin is good at detection of heteroresistance. For oxacillin, if we use oxacillin along with cefoxytin, it may be used for the detection of BORSA also, because in BORSA it will be cefoxytin susceptible but oxa resistant, and we have to follow the time of the incubation also very, very strictly. When it comes to table 3H of the M100 document, the oxacillin salt agar test. So in this case, we are using 4% NaCl. We are using 4% NaCl, and the amount of oxacillin is 6 microgram per ml. So we don't use the disc diffusion, we use the oxacillin screen agar. Again, in this case, the incubation is done for 24 hours, and it is read with transmitted light. So whenever you are suspecting the heteroresistance, so you have to look at finer colonies and the small colonies which are growing. So that is why we have to use the transmitted light and not the reflected light. So there are different MRSA chromogenic media which are available in the market. This is denim blue agar by Oxide. This is by Biorad. This is by Biomiro, Becton Dickinson. So they are all available in the market. Personally, I have not used any of them. There are some very interesting questions that why is 4% salt added? So I already mentioned, for the enhancement of the expression of the beta-lactamase and the MEK gene. Why we use oxacillin to detect methicillin-resistant Staph aureus MRSA? Because physiologically, oxacillin is more stable than methicillin. Why not use oxacillin disc instead of, and why oxacillin agar only? And again, why use cefoxytin disc instead of oxacillin disc? So the answers are the same. That has been provided by the UCAS document that was released in 2017, detection of by MIC, determination or distribution. So they say, this is a UCAS. They say that the heterogeneous expression of resistance particularly affects the MICs of oxacillin, which can appear susceptible. Cefoxytin is very sensitive and specific marker of MEC and MEC mediated methicillin resistance, including in heterogeneous expression strains, and it is the agent of choice. This diffusion using oxacillin is discouraged, and interpretive zone diameters are no longer included in the UCAS breakpoint table, and these are also eliminated from the CLSI. But CLSI does not give this kind of a explicit explanation, this kind of like that. So why oxacillin and cefoxytin are tested instead of methicillin? So most of you might be knowing answers, because methicillin is now no longer commercially available, and methicillin was less stable as compared to oxacillin. But why cefoxytin is better is that because cefoxytin is more likely to detect the heteroresistant strains. Cefoxytin is the better inducer of the MEK gene. Cefoxytin also gives more reproducible and accurate results as compared to oxacillin. That's the reason why we don't require to read it through a transmitted light. We just need the reflected light in such cases. Cefoxytin also gives much clearer endpoints, thus, and it is the preferred method for all the SOSA isolate, that is the Staphylococcus other than Staph aureus. So if it is an ideal world, we should use the cefoxytin disc and oxacillin MIC, ideally by BMD method, and also the nitrosephin method, especially for the BORSA isolates, the penicillin disc, of course, and the amoxiclav again to detect the BORSA isolates. And also, of course, check for the MEK with PCR, MEC with PCR, MEK with latex agglutination, or penicillin binding to A by the latex agglutination. Is it oxacillin and methicillin resistance? Yes, because like I said, heteroresistance is very, very common in Staph aureus because of multiple subpopulations. One might be susceptible, one might be resistant, and they are coexisting with each other because all the cells are carrying the MEK gene, and there's only partial expression of the gene. So that's why the heteroresistance is there, and it's very difficult to detect. And especially in cases of oxacillin, we are seeing heteroresistance cannot be detected very well with oxacillin, and that's why it requires cefoxytin, because that evenly expresses the MEK expression. So cells which are expressing the heteroresistance, they grow more slowly than the oxacillin susceptible populations and may be missed at temperatures above 35 degrees. So that's why if you see, all the temperatures for the gram positives are at 35 degrees, especially for Staph aureus. And that's why the temperature is maintained at around 33 to 35 degrees, not 37 degrees, for a full 24 hours because the cells are growing slow. That's why we must follow the time of incubation very, very carefully. So you should note down the time of the incubation also, especially if you're detecting the MRSA by the oxacillin methods. Few more questions. Oxacillin and cephalosporin, why the isolate is called MRSA? Purely for historical reasons, just nothing like that. How do MRSA, anti-MRSA beta-lactams act? So we know that the anti-MRSA beta-lactams are ceftaroline and ceftabiprole. For some reason, the ceftabiprole, the word ceftabiprole has not been mentioned in CLSI anywhere. They are mentioning only ceftaroline, which is a fifth generation cephalosporin. Recently, I read that some of the ceftabiprole trials are still going on. So probably that's why they're not still included as a therapeutic agent in CLSI. It is normally the beta lactams don't bind to the penicillin binding PBP2A. Now, what happens is the PBP2A is protected from the beta lactam drugs, which is normally present in an inaccessible group. What ceftaroline does is it binds to the allosteric site and changes the structure of the penicillin binding to PBP2 protein and opens the active site on the PBP2A for the second molecule of ceftaroline to bind. So basically, it creates a allosteric change in the altered penicillin binding proteins. It changes the structure of the PBP2, and further on top of that, it adds itself to the groove, and it inhibits the transpeptidase activity of the PBP2A. This is just a report of MRSA isolate from our institute. This is from a surgery patient, inpatient. In this case, we isolated an MRSA isolate, and we have advised teicoplanin as a drug of choice, and we have released vancomycin, but we have advised teicoplanin as a drug of choice. It is inducible clindamycin resistance negative, ICR negative. Why we have given? Because I'll just tell you this. I think we'll be repeating this report later on also. In this case, as you can see, that we are advised teicoplanin in this case because we are doing the breakpoint to MIC quotient reporting also, and the susceptibility breakpoint of teicoplanin. I'll just show you. I think it's gone previously. No, I'll think, yeah, it'll be come. It'll be coming. So, so why is it important for the clinicians to know about MRSA? Because MRSA is not the same as the normal Staph aureus. It has got resistance to multiple drugs. It is quite fulminant, especially the community-acquired cases. They may cause the expression of the PVL toxin, for which clindamycin or linezolid may be required. The clindamycin and linezolid both are protein synthesis inhibitors, and they are good at suppressing the expression of the. So linezolid per se has got a very good activity, not only as the anti-MRSA drug, but also in suppressing the activity of the toxin also. And so severe infections have been found, like necrotizing pneumonia, septic. So they can produce a lot of severe infections. So that's why we have to know the in and out of MRSA also. So community-acquired MRSA and the hospital-acquired MRSA have got different risk factors, like sport participants, inmates in correctional. So all these things should be present, should be taken in the patient's history. So what is the background of the patient? Who is the patient is, where is he coming from? And the risk factors include the contact, crowding, contaminated items, compromised skin integrity, and absence of cleanliness. So these are risk factors for the community-acquired MRSA. Similarly, we got risk factors for the hospital-acquired MRSA also, like the prolonged hospital stay or the care in the ICU, prolonged antimicrobial therapy, multiple drugs, surgical procedures, close contact with infected patients, chronic medical risk factors are there for the hospital-acquired MRSAs also. Coming to the resistance to glycopeptide drugs in Staph aureus, we'll begin with the vancomycin susceptible MRSA, where the vancomycin MICs are less than equal to 2, the heteroresistance visa, the visa, and the vancomycin resistant Staph aureus. So I said initially that when vancomycin was discovered in the 1960s, it was in a very crude form. It was a red-colored powder, and it used to be known as the Mississippi mud because of the crude red color. And it had a lot of toxicities. And even now, they've not been able to refine the nephrotoxicity by vancomycin. It binds with high affinity to the penultimate D-alanine residues or the newly synthesized pentamers, which are causing a disruption of the peptidoglycan assembly. So they have got actions. They are also cell wall active agents, just like the beta-lactam drugs. So we back to the table 2C, and this is why I was referring to the previous report of MRSA, why we are advising teicoplanin. So now in this case, vancomycin MIC, as you can see, there are no breakpoint zones, right? So there are no break for the lipoglycopeptides. There are no breakpoint zones for the teico breakpoint zones for vancomycin. There are only MICs available. So please don't use discs of vancomycin. Don't use discs of teicoplanin also, except maybe in case where the breakpoint zones are available for, like in case for Enterococcus. So if you see the breakpoint MIC, that means the MIC below which the isolate is likely to respond clinically, that is 2 or less than 2 in case of vancomycin, whereas in case of teicoplanin, which is still investigational as compared to CLSI, and CLSI is an American document, mind that CLSI still writes it as investigational because not approved by for the clinical use in the USA because it's not still FDA cleared. So why we are insisting on teicoplanin is because the breakpoint to MIC ratio, because you see the susceptibility breakpoint of teicoplanin is much higher as compared to the vancomycin, and therefore even we get the MICs also of teicoplanin much lower. So the ratio of the susceptible breakpoint MICs, susceptible breakpoint to the MICs obtained is much higher in case of teicoplanin as compared to vancomycin. So again, just a reminder: do not use the vancomycin discs for staph Staph aureus or for staph other than Staphylococcus aureus. Now, what's this point number 15 is very interesting. For Staph aureus, vancomycin susceptible isolates may become vancomycin intermediate during the course of prolonged therapy. So, if you are suspecting a patient is not responding or they is taking a prolonged time to respond to vancomycin therapy, you have to go back and see the history of vancomycin intake. And also, FYI, vancomycin should not be given as a monotherapy. Vancomycin should, for MRSA, always has to be supplemented with some other drug because vancomycin is a molecule. It's a very large molecule, and that's the reason why we don't have breakpoint zones, because a breakpoint of 18 is susceptible and 17 is resistant has got a margin of only 1 millimeter. So there can be a lot of errors when it comes to the breakpoint zone calculations for vancomycin. That's why it is better to have MICs only. Now, coming back to the same report of MRSA, if you can see over here, this is what I was saying, that the breakpoint MIC, susceptible breakpoint MIC is 8 for teicoplanin, for vancomycin is 2. But the MIC of the isolate that we obtained is 0.5 and 1.5 for vancomycin. This is by e-test. So you can see MIC by agar gradient method. This is the actual screenshot of our AST report. This is not that I've made something specifically. This is a random report of an MRSA that I've picked up. So the d-test is negative for inducible clindamycin resistance. And in this case, we have also even given the dosing of the antibiotic as per the John Hopkins guidelines and as per the Sanford guidelines that we follow. The septic arthritis, heavy inoculum infection, we give septic if it is an asymptomatic infection or a non or a local infection, what to do. If d-test is negative, clindamycin may be added if signs of toxemia are present to neutralize the effect of toxins. So all these notes are going on our AST reports. Coming to the MIC creep that we have, that's a very common phenomenon that we see in vancomycin, that patients infected with vancomycin with MRSA, they experience a phenomenon known as the MIC creep. They experience poor clinical outcomes, there is delayed in treatment response, there is increased mortality, there's increased rate of relapse, extended hospitalization, and increased cost also. So as you saw in that table, point number 15, the patients who have taken prolonged vancomycin therapy are likely to have, or they are like to get a MIC creep, and the MIC creep will depend on the type of the AST method. So what actually happens during MIC creep is because the constant exposure of the antibiotic, the cell wall starts thickening. That is the reason of the development of the H-visa and the visa isolates, because of the thickening of the cell wall. So it also depends on the type of the Staph aureus strain, the type of patient and the population evaluated, and of course, also depends on the type of AST method used. So in some of the surveillance projects and service program, many people are reporting vancomycin MICs of 4 and 8 without even confirming. So please be very, very careful before you're reporting any vancomycin MIC of more than 2. So it is not a very, very good method for reporting MICs. You have to counter check, you have to cross check your quality control using the ATCC 29213. 29212 is Enterococcus, 29213 is Staph aureus, that is used for MIC detection. So coming to table number 3I, that is the vancomycin screen agar for the Staphylococcus. Again, this is 6 microgram per ml of vancomycin, and for this we have to do inoculation of 0.5 McFarlane turbidity standard, and using a micropipette we have to do a spot inoculation of 10 microliter drop onto agar surface, and then incubate at 35 degrees for 24 hours. If there's a growth, then there's a presence of probably vancomycin. So we have to confirm. If it growth is there, you have to confirm. We just don't blindly give it as vancomycin intermediate Staph aureus or vancomycin resistant. We have to first confirm it. So this is the old protocol of CDC where the vancomycin disc zone. So this is not, but the basic protocol is quite similar, that they used to initially screen and then do the MIC detection using a vancomycin screen plate and also the BMD methods, and then check for purity, to confirm the ID, and then send the isolate to a referral lab for confirmation of the vancomycin resistance. Coming to the VISA isolates, the vancomycin intermediate susceptible Staph aureus isolates. This is a 2015 document of CDC that tells us that ideally we should use the reference broth microdilution method for the detection of the VISA isolates. So if the MIC is more than 16, vancomycin resistant, but if it is between 4 to 8, then it is most likely a VISA. But the important point is that we have to give a full 24-hour incubation time because of the heteroresistance, which is also present in the in glycopeptide resistance also. Then there's commercially prepared Brainhard infusion agar containing 6 microgram of vancomycin. If we see a colony of more than one colony in 24 hours, then we have to confirm again with the broth microdilution also. Of course, there's gradient E test also, but gradient E, keep in mind, it normally gives you 1 to 2 log dilutions higher as compared to the broth microdilution. So whenever you're noting down your values of MICs, please write down MIC BMD or MIC E test like that. So that there's no confusion, because if it is MIC BMD, then the values of the MIC BMD would be slightly lower as compared to the MIC E test. So what is VISA? VISA basically, it has got reduced susceptibility to vancomycin, but there's no frank resistance. And it is not due to the acquired antibiotic resistance gene, but it results from the accumulation of mutations. So what happens is that there are multiple mutations that keep on accumulating, and it begins increased thickness of the cell wall. So the ones which have got only increased thickness of cell wall, they are normally labeled as H-VISA, and which are the outcomes of the MIC creep. Other than that, there are additional mutational changes like the cell wall changes leading to anomalous division of vancomycin, diffusion of vancomycin through the VISA cell wall, decreased negative cell surface charge and less vancomycin binding, decreased autolysis, increased cell wall synthesis, and decreased peptidoglycan cross-linking result. High affinity binding of vanco to non-aminated neuropeptides. So these are the changes besides the thickening of the cell wall. So the thickening of cell wall is only change that creates the H-VISA, whereas the further mutational changes leads to the development of the VISA isolate. So if we talk about the H-VISA and all these things, so H-VISA is the heteroresistant VISA that develops into a frank VISA later on. So similar to MRSA, the VISA phenotypes is commonly preceded by a hetero and multiple mutations in the H-VISA strains lead VISA phenotype. The mu3 was the first H-VISA that was characterized, which was proven to have a thickened cell wall and not the presence of other mutations. Like the hetero MRSA, H-VISA also reveals uneven killing of a seemingly homogeneous cell population, and there is no cross-resistance to teicoplanin. So you can see the rise in the MIC to vancomycin, but not to teicoplanin. In H-VISA, majority cells have no resistance to vancomycin and they get killed by 2 microgram per ml. But there is also a subpopulation that is existing that survives vancomycin concentration of 4, and they start behaving visa strains. So basically, H visa is a combined population of the vancomycin susceptible Staph aureus along with the vancomycin intermediate Staph aureus. So it's a combined hetero or a combined subpopulation of these isolates. In the H visa, like I said, there's only thickening of the cell wall is happening in the absence of mutations associated with the visa strain. So H visa is only the thickening of the cell wall, and visa is the additional mutations that add on to the H visa to create a higher MIC for vancomycin. Coming to the slow H visa, it is characterized by a and it requires 72 hours of growth or more for colony formation. It has got relatively high MICs of almost 8 to vancomycin compared from visa. It has got an unstable profile and they revert back in the absence of vancomycin. Mutations in the RPOB and the RPOC genes of the RNA polymerase, they are the reason for the slow visa to emerge. And lower concentration mupirocin. It's very interesting that lower concentrations of mupirocin enable the isolation of stable S visa strains, and this approach is used to demonstrate the presence of S visa isolates. Coming to the frank MRSA, the frank VRSA. VRSA carries the VAN A operon. So this VAN A is very similar to the same that we see in the VAN A of Enterococcus also. So VRSA carries the VAN A operon that provides the cell with two important activities. One is the hydrolysis of the existing D-alanine precursors. So D-alanine precursors are the normal precursors which are the components of the cell wall. In that case, the normal precursors get hydrolyzed, and the new ones they get synthesized, D-alanine and the D-lactate precursor, which is vancomycin. Now, I'll just in the later slides also you'll see the D-alanine and the D-lactate and the D-alanine and the D-serine. They're two different kind of altered precursors in which the D-alanine, D-lactate, they cause the high level resistance to vancomycin, and the D-alanine and the D-serine cause the low level resistance to vancomycin. The first case of VRSA, frank VRSA, was reported in 2002 in US, and then it was found that this particular strain it coexists co-infection with an Enterococcus fecalis isolate, and this MRSA strain acquired the VAN A operon from the conjugative E fecalis plasmid. So that means there is an interspecies transmission of plasmid also that is conferring resistance to the vancomycin through the VAN A operon, and this causes a MIC surge of more than 1000 microgram per ml. So there are very few cases because it is not very healthy for the VAN A gene to be present inside the MRSA for some reason. The fitness of the plasmid is not very considered to be very good inside the MRSA, and because of this, the plasmid does not survive for a long time and is either lost or it gets destroyed. So the vancomycin isolates, the MRSA strains, the incompatibility with methicillin resistance because of the MEK encoded transpeptidase. That's the reason for this, the absence of vancomycin resistance and MRSA together. So this is a seesaw effect you can see in this case. Coming to the resistance to the macrolides. Just a minute. Okay, coming to the resistance of the macrolides. That is the macrolide lincosamides, streptogramins, and MLSB resistance. That is commonly called the D-test or the inducible clindamycin resistance. So coming to table 3J, that tells us that how we're supposed to do the distribution testing for D-testing. In this case, very interesting thing is that we have to include only those organisms which are resistant to erythromycin and susceptible or intermediate to clindamycin. So this holds true for all Staphylococci and for beta H's, that Streptococcus. So for this, what we need to do is for the Staph, we just need a regular MHA, or we for Streptococcus pneumoniae, of course, we require the MHA supplemented with 5% sheep blood. So please keep in mind one very important thing is that we are not supposed to use sheep blood agar here. We are supposed to use MHA supplemented with 5% sheep blood. So this is a very common error that we are all doing on a daily basis, that we are using sheep blood agar. If you're making sheep blood agar and you're making it in a MHA base, that's fine. But then otherwise, plain simple sheep blood agar with Columbia blood agar base, maybe that's not a very good option for detection of B test. So anyway, so in this case, we are supposed to keep the erythro and clindamycin discs together. Now the important part is it is 15 to 26 millimeters edge to edge for Staph aureus, whereas in case of Streptococcus pneumoniae and the beta HS, it is 12 millimeters apart edge to edge. So please keep in mind, you'll have to tell your technical staff also the distance that you are using between the two discs is 12 millimeters edge to edge for Streptococcus pneumoniae and for the beta monotic strains, whereas it may range from 15 to 26 millimeters apart edge to edge in case of Staphylococcus species. So we have all seen this thing. It's a very common thing that we are observing, the D test. So the important part is that clindamycin per se may not exert its antimicrobial effect, but it has got a very good protein synthesis inhibitor. So it has got a good antitoxin effect, especially if given in a dose of 600 milligram TDS. Uh, Even though there are non-FDA approved uses for clindamycin, but it is shown to decrease the toxin production caused by MRSA, Streptococcus pyogenes, and even Clostridium perfringens. So if you're suspecting a very fulminant case or a clinical case where there's a lot of toxemia is there, the lot of toxin production could be present because of the patient's clinical condition. Patient is very sick, moribund, and there is Staph aureus infection. So such cases are likely to be producing community-acquired MRSA with a lot of PVL toxin being produced, and clindamycin in such case, and even linezolid will neutralize the toxin. Coming to the resistance to the oxazolidinone drugs in Staphylococcus aureus, coming to the linezolid resistance, you've got chromosomal point mutations, and which are also, there are some plasmid mediated resistances also. Linezolid is a very, very good drug. It is orally available and almost 100% oral bioavailability is there. It's the second most frequently abused antibiotic after augmentin. And it binds to the 50S subunit of the bacterial ribosome through the interaction with the 23S rRNA and blocks the protein synthesis. So it's a protein synthesis blocker. So like I said, because the PVL toxin is also a protein, and therefore linezolid has got a good action in reducing the toxin production, just like clindamycin. So the resistance is associated with the distinct nucleotide substitution. So what is happening is that usually their point mutations are like this. So your G2447T is, that means at position G at 2447 replaced by thymine. It's like that. So especially like this is a very common mutation, G2576T. This is a very common mutation that's found. And we've also seen that the linezolid resistance is much more common in Staphylococci other than Staph aureus. So we have had multiple publications regarding this. And this publication, we had the detection of the classical G2576U mutation in the linezolid resistant Staph aureus, because U is in the RNA. So that's why we written U over here. With the DNA, it will be T over here. So we found a co-resistance of linezolid in Staph aureus as well as in Enterococcus also. And then the 23S rRNA, the 23, we did the PCR for the 23S rRNA, that particular domain, it creates a amplicon of around 746 base pairs. And then 746 base pair amplicon was sequenced by Sanger sequencing, and we found a point mutation at the G2576U position, which is the domain 5 of the 23S rRNA. So there is a single point mutation happens, and this single point mutation is the commonest reason for the resistance towards the linezolid. Then another study that we did was that the detection of the cryptic resistance. What is cryptic resistance? Cryptic resistance means hidden resistance. What happens is that 23S rRNA has got multiple alleles which are present. So almost 5 to 6 different alleles are present in case of Staph aureus. 6 alleles are present in Enterococcus faecium, I think. And it depends on how many alleles they get mutated to produce a frank resistance. So before the production of the frank resistance, there might be 1 or 2 alleles which might get mutated and they are not expressed phenotypically. So resistance. So what we did was we tried to find out what are the hidden resistances over here. And when we did this particular study, we found at least 2 isolates of MRSA which were linezolid susceptible, but they did contain the hidden resistance or the cryptic resistance. So if you see over here, PCR was done for the domain 5 of the 23S rRNA that yielded a 746 base pair amplicon. Now what happens is that this particular single point mutation, it creates a restriction site. That means it is active for the restriction endonuclease, that is 1. That NHE1, it cuts the amplicon at that particular point, and it splits the amplicon into two separate bands of 557 base pair and 189 base pair. So this 746 band will be present in all the gram positives. It will be present in all the gram positives, but the if we do the either we do the Sanger sequencing or we do the SNP, the single nucleotide polymorphism, or the PCR-RFLP, if you do, we'll find out that only the ones which have got a single point mutation, the restriction site will be created, and after the addition of NHE1, it cuts the amplicon into the 189 and the 557 base pair. Thereby, when we run the gel like this, so it will show you two separate bands. So this is a very old technique. Yes, it's been now replaced with WGS, but still, for the purpose of knowing what is the conventional PCR, we must know this. Then we have got a plasmid-mediated resistance towards linezolid that is mediated through a ribosomal methyl transferase CFR gene, which is located in the plasmid and other mobile genetic elements also. It is also present in the commensal flora like this, the Staph aureus and Staph aureus, and can be transferred to the clinical MRSA isolates. One good thing is that linezolid may show resistance, but tedizolid remains active against the CFR gene resistance mechanism, and it is 4 to 6 times, 16 times more potent than linezolid, and much more dramatic fall in toxin production can be seen. So if we see there is linezolid resistance, maybe we can go in for a tedizolid treatment also if possible. Now, small colony variants are also very commonly missed causes of drug resistance, and these are basically nutritionally deficient staph isolates which have been exposed to multiple times, multiple antibiotics, and they become they are easily missed mutant strains, and they are known to cause recurrent infections, and they harbor atypical phenotypic and resistance pattern. And they've got a very uncharacteristic appearance on sheep blood agar, and we are likely to miss them. And that's why we need to reincubate the plates. If we see tiny colonies, we need to reincubate. And these are the kind of, we can say, oxotrophic strains that arise out of chronic exposure to antibiotics. So this was a very interesting case that looked like a gram positive, like a, you can say, a diphtheroid growth from a aspirate from a patient's lymph node. And we found very, very tiny colonies. When we made the gram stain, we found this gram positive cocci. So just out of curiosity, we incubated it, and after multiple days of incubation of 3 days to 4 days, we found it classical, the egg, the fried egg colonies of the small colony variants of Staphylococcus aureus. We published this case also. This is a very interesting case. So these are the small colony variants. They cause recurrent infections. They cause repeated infections. And why? Because we are easily missing on them, because they're very tiny. They would show very atypical growth on blood agar, and they may not grow on McConkey at all, and they may also not exhibit the classical features like the tube coagulation. Then there is mupirocin resistance also in the gram positives. So there is mupirocin resistance, can be of low level or can be of high level. That also shown in the CLSI also. This is also another case of a patient who was colonized with mupirocin resistant MRSA, and we failed to decolonize it despite the isolate being susceptible to erythromycin. So similarly, in this case also, we did the PCR RFLP, and we found that the mutation was present. And in table 3K, that tests the mupirocin disc using the 200 microgram mupirocin disc, and again 24 hours incubation time, and read with transmitted light, we see the growth. It is typically for the high level mupirocin resistance, that is more than MICs of more than 512 microgram per ml. Coming to drug resistance in Enterococcus, there is resistance to amino-glycosides, glycopeptides, resistance to oxazolidinones, which is almost same as Staph aureus, and resistance to the beta-lactam drugs. So this one, it tests about the HLAR, so the high-level aminoglycoside resistance. So we are testing, CLSI recommends 2 antibiotics, that is gentamicin and streptomycin. So gentamicin, we are using gentamicin 120 and streptomycin 300 discs. Ideally, both of them should be used, but if you got deficiency or some issues are there, so only gentamicin can be used, because gentamicin would be covering most of the aminoglycoside modifying enzymes. So the most common cause of the aminoglycoside resistance in Enterococcus is the aminoglycoside modifying enzymes. Other than that, there are intrinsic resistances also to aminoglycosides, because the entry of aminoglycosides inside the Enterococcus is oxygen dependent. the cell wall is very difficult. It does not penetrate. So normally at usual concentrations or the levels which are usually achieved in the blood, these antibiotics cannot affect minoglycosides cannot affect Enterococcus, and therefore for that they are always combined with the cell wall active agents like the beta-lactam drugs. Until unless the beta-lactam drugs don't break the cell wall, they cannot enter inside. So that's why they are never given as monotherapy for Enterococcus. And overall, generally also, aminoglycosides are never to be given as monotherapy. They are not to be attempted as treatment of choice for lung infections because they don't achieve very good concentration. They monotherapy for some of the urinary tract infections because aminoglycosides have very good concentration in the urine, so they can be given rarely as the monotherapy for lower UTIs, especially caused by NDMs, in which case many of them are only amica susceptible and resistant to all antibiotics. So if you see that there is resistance in the gentamicin or in streptomycin, in that case, it is not synergistic with cell wall active agents like ampicillin, penicillin, and vancomycin. So in such cases, you cannot give this treatment together. Now, why we are using only gentamicin and streptomycin? Because other aminoglycosides don't need to be tested because their activities again Enterococcus are not superior to gentamicin and streptomycin. That is one thing, because genta and strepto, both of them together, they cover almost all the different aminoglycoside modifying enzymes. A very interesting information I wanted to share is that I think many of the examiners may also ask that some of the antibiotics, the aminoglycosides are ending with mycin, MYCAN, and some are ending with MICAN. So what's the difference? So the aminoglycosides that are derived from bacteria of the Streptomyces are named MYCIN, whereas those which are derived from Micromonospora are named with the suffix of MICIN. So that's why we got tobramycin as MYCIN and gentamicin as MICIN. So please be careful about the spellings of these aminoglycosides also. Like I said, some of them are intrinsically resistant to the aminoglycosides, and intrinsic enzyme mediated HLAR, it is not described for genta and strepto. So if you see that, let us say if we start using tobramycin, to get the HLAR, so some of these strains, especially Enterococcus faecium, they show intrinsic resistance to the to tobramycin. So in that case, no point of using it. Gentamicin and streptomycin, they are the only two antibiotics for which there is no intrinsic resistance to aminoglycosides has been described for Enterococcus, and both they retain the synergism with beta-lactams and they cover all the aminoglycoside modifying enzymes. So if you see this table, the aminoglycosides per se have got a low cell wall permeability. They do not penetrate the cell wall of the Enterococcus solely, and that is why cell wall active agent like beta-lactam drugs or vancomycin. That's why we use the term high-level aminoglycoside resistance, HLAR, and not the usual ones. Also, there are a lot of aminoglycoside modifying enzyme which show intrinsic resistance in case of Enterococcus faecium. Like I told you, tobramycin, kanamycin, they show intrinsic resistance in case of Enterococcus faecium, so they cannot be used for HLAR detection. And there are numerous other enzymes which are aminoglycoside modifying enzymes which are more frequent in Enterococcus faecium as compared to Enterococcus faecalis. Then we also know that the Enterococcus are intrinsically resistant to many of the cephalosporins. They are resistant to clindamycin. They are resistant to chloramphenicol also. So this is why we have to be careful that we should know that what are the penicillin binding proteins which are to which the beta-lactam drugs it's intrinsically resistant to. Like for example, PP4 predominantly seen in Enterococcus faecalis, PP5. So these penicillin binding proteins, they don't get bind to cephalosporins. They don't get bound to carbapenems also. So that's why there is intrinsic resistance is there. So normally they don't act. They might show in vitro susceptibility, but in vivo they act very well. And like I said, beta-lactamase in Enterococcus is very rare. It's not very commonly found. If found, rarely in E. faecalis only it's there, and that too the lactamase gene same as Staphylococcus, that is the BLA-Z gene. The appendix B. Now, the appendix B is very, very important. Appendix B of CLSI shows you what are the intrinsic resistances. So you must know the Staphylococci are intrinsically resistant to many of the antibiotics like Astronam, polymyxin B, colistin, and nalidixic acid. Similarly, the Enterococcus the appendix B for Enterococcus is also very interesting. This is your clear-cut warning that for Enterococcus species, cephalosporins are minoglycosides. Except for high-level resistant testing, clindamycin, cotrimoxazole may appear active in vitro but are not effective clinically and should not be reported as susceptible. Now, the interesting point is that most of you who are doing the quality control of MHA would be putting up this. You would be putting up a cotrimoxazole disc against the ATCC 29212, that is Enterococcus fecalis, for quality control of MHA agar. So why do we do that? Why are we testing cotri when we say that cotri is resistant? Why are we testing? So the purpose of doing that is that Enterococci, they are unique. They are very different. Normally, what happens, the bacteria, they are not utilizing folic acid from the medium. They have to synthesize. So their folic acid synthesis pathways are there. And cotrimoxazole, it inhibits the folic acid synthesis pathways. But Enterococci are unique in the sense that they can absorb the folic acid directly from the environment, bypassing the effect of cotrimoxazole. Therefore, if your media contains folic acid, then it will show resistance to cotrimoxazole. But if it does not contain folic acid, does not contain thymidine residues, in that case, it will show susceptibility. So that means your media, like MHA, has to contain low level or zero level of thymidine, zero level of folic acid, and that's why we test cotrimoxazole in ATCC 29212 in case of. Coming to the resistance of glycopeptides, there are primarily three types. That's the VAN A, VAN B, and the VAN C phenotype. The VAN A phenotype shows resistance to both vancomycin and teicoplanin, and it is found in Enterococcus faecium and fecalis, both of them. It is acquired kind of resistance. In VAN B you see resistance only in vancomycin, teicoplanin. You see an intrinsic resistance to both vancomycin and teicoplanin. So if I just go back to this table of appendix B, you see over here, Enterococcus gallinarum and Enterococcus cassiflavus is showing resistance to vancomycin and not to teicoplanin. So this is the intrinsic or the inherent VAN C resistance is present over here. So like I said previously, the usual binding site for vancomycin are the D-alanine sites, for which the vancomycin MIC are usually less than or equal to 2. In case of low-level glycopeptide resistant enterococci, GRE means glycopeptide resistant enterococci, the D-alanine, D-serine combination is there because of the mutation, and this causes the vancomycin increase MIC to more than between 4 to 32 microgram per ml. But in case of high-level glycopeptide resistant enterococcus, what we commonly see in the VRE, we have the D-alanine and D-lactose replacement is there, and which the vancomycin MICs are even more higher. In the end, coming to the resistance in pneumococcus also, like Streptococcus pneumoniae, was initially quite well dealt with penicillin, and they had the penicillin-resistant Streptococcus pneumoniae, for which they developed the cephalosporin. Then cephalosporin resistance also came. Levofloxacin, moxifloxacin, they are sparfloxacin, they are very good respiratory fluoroquinolones. Again, for respiratory fluoroquinolones also, this started coming. Main reason being the Streptococcus pneumoniae. For a person like me, even today, dealing with the respiratory pathogens or dealing with the respiratory samples is very, very difficult. The Streptococcus pneumoniae, which I'm getting in a throat swab, is that a true pathogen or that is a commensal flora? We don't know. Many of the humans, we are carrying the Streptococcus pneumoniae in the throat or maybe in the oropharynx, and we are getting exposed to a lot of fluoroquinolones. That is one of the reasons why the acquisition of fluoroquinolone resistance in the Streptococcus pneumoniae. So we have to know how to differentiate between the true pathogen and the commensal flora. So that's very difficult at times in case of the respiratory samples. Then once the vaccination was reduced, so we also had the emergence of the MDR Streptococcus pneumoniae also. So the main resistance that we see in Streptococcus pneumoniae are resistance to beta-lactam drugs, where we see the alteration in the cell wall penicillin binding proteins. We see the resistance in macrolides because of target site alteration by the enzyme that methylates 23S rRNA subunits through the ERMB mutation. And of course, the fluoroquinolone resistance is also there because of spontaneous mutations in the quinolone resistance determinant region. So this QRDR resistance mechanism is also common in Salmonella typhi also. So these are the major resistance mechanisms and the methods of their detection in Staphylococcus. Enterococcus, and in Streptococcus pneumoniae. So, at the take-home message I would like to give to the attendees is study and imbibe the basics to the most advanced, because we are a generation that have to learn from coagulase to MALDI. We have to read good articles, all the articles from ASM, like anti agents in chemotherapy, Journal of Clinical Microbiology reviews, all the mini reviews, reviews, standard operating procedures from ASM, the Qmitex from ASM, journals like Journal of Antimicrobial Chemotherapy, Clinical Infectious Diseases, Journal of Infectious Diseases, uh, infection control, hospital epidemiology, of course, our very own beloved journal, Indian Journal of Epidemiology, American Journal of Infection Control, British Society of Antimicrobial Chemotherapy documents. So these are the sources that we have to read on a daily basis whenever and wherever possible, because we just can't rely on 3 or 4 books. We have to go through all these. And of course, the must-read documents like UCAS. UCAS has got all free open sources documents and all the rational documents that you can go through. CLSI M100, M39, M2, M7. These are the four, I would say, the four Vedas of AST that we have to read. Desirable ones are M45 for the fastidious organisms and M11 for the anaerobes. CLSI newsletters and rational documents, CDC guidelines and MDROs, the IDSA and the American Society for Healthcare Pharmacists guidelines and guidance document whenever and wherever possible. With that, I'd like to end this masterclass with a note of thanks to Dr. Sonal and the entire team of Indian Journal for IAMM also. Thank you so much, Dr. Sonal.
Thank you, Sumit. That was excellent. And there are a few questions. And then I will ask Bharti ma'am, who joined, to wind up. One of the question is that we say that the vanco resistance has to be given, intermediate is between 4 to 8. So why do we use a vanco screen agar with 6 micrograms per ml?
We are using 6 microgram per ml because we have to detect the entire variate range. Actually, the range will start from 4 to 2. So anything that is growing between that, so we have to cover the entire range. So ideally, maybe in the future, they might get rid of this, because one single breakpoint is not sufficient to detect the entire range of the VISA isolates. So yes, it is a very valid question. I cannot really give you the rationale why only single this thing is used. The agar dilution method should be used for vancomycin VISA
Yes,
detection.
because this vanco screen agar also misses VISA. So once we have more of VISA coming in, I think we'll have to change the methodology or
Yes.
change the concentration. Right now, VISA strains are so few in the world that we really are not bothered by that small little number. And probably
Yes.
it is that if you increase more vancomycin concentration in the agar medium, the medium becomes very thick. So
Yes.
the growth of the organism per se becomes very, very difficult. It eats up the moisture kind of thing. So that is why we could not use disc also, because the diffusion is so slow. So
Yeah.
I think that could be one of the reasons that they standardized it at 6 maybe.
Yes.
So this was one. Another question is that you said MODSA mimics MRSA phenotypically, but can we differentiate between the two phenotypically, that is MODSA and MRSA? And secondly, does the treatment differ between the two strains and the clinical implications of this in the light of MRSA infections?
So the MRSA isolates are the isolates which do have the PPP2 mutation is there, but they are unconventional mutations. They will be negative for the MEC-A. So we need to do maybe a latex agglutination or a PCR for MEC-A. If they are negative, but they are cefoxitin resistant and they are resistant to oxacillin, then in that case we can label them as MRSA.
Yes.
Treatment should not vary because these are also altered penicillin binding proteins. So in that case, ultimately you may have to go in for a glycopeptide along with some other antibiotics. So glycopeptide therapy should not be given as a monotherapy.
All right. Now, people have asked about sharing of CLSI document. Now, I'll answer that on behalf of Sumit. CLSI are patented documents which are purchased, and they should not be shared even on WhatsApp and other social media or on the platform. But if you want to really read CLSI, they are available for reading purposes if you log in and create your account in CLSI, which is valid for 1 full year. So you can read there. You cannot select anything, save anything, maybe you can click a photo, but you cannot download anything from their website, but you can read now. That means your generation is that much lucky. We didn't have that also. So and the other is that all those who have access to WhoNet, if you update your WhoNet, the whole thing automatically gets updated to the latest CLSI. So that also gives you another this thing. And again, one more question: that any implication for treatment of community-acquired and healthcare-acquired MRSA? Sumit, I think that will be the last question that we get.
Ultimately, the entire treatment cannot be given as a blanket therapy. We have to rely on the AST. So what we do in our case, like we have to first see if the patient is from OPD or from the. If the patient is from the OPD and he's got we assume that that's a community-acquired MRSA. In that case, we look at all the oral antibiotics and we release the oral antibiotics and we try to avoid the parenterals like vancomycin and teicoplanin. So we do get a very good, at least in our setup, we get a very good susceptibility to doxycycline. We get a very good susceptibility to chloramphenicol, to cotrimoxazole. So we do release the oral antibiotics for the OPD patients, which are the query community acquired MRSAs.
All right. So I think we've already crossed our time limit. So I will request Dr. Bharti, our president, ma'am, to please say a few words and wind up this discussion.
So thanks a lot, Sumit. That was really exhaustive presentation, and you really explained beautifully all aspects, whether it be the pathogenesis or it be the lab and the clinical aspect, very well, compiled together. So really thanks a lot. And I think you have a huge audience to that because of your reputation, and really that was a great presentation. And how beautifully you describe what the visa and the VRSA and all those strains, how to diagnose them and where we can go wrong. And the best part, I think, the carry home message, all the material you provided, where to read from, will really help the students. So I think I'll just cut it short. Thanks a lot for your presentation, and we look forward to more such presentations from you.
Thank you, ma'am. Thank you.
Thank you, ma'am, and thank you everyone for joining in. Thank you so much.
Thank you.
Thank you.